I think actually I want this. All right. The final is comprehensive, but about 50% of the final will be on material you have not been quizzed on previously. So 50% of the material will be from chapter 14, chapter 13, and then the unknown project. There will be no questions about the infectious disease project because each of you are working on an independent and separate disease. Any questions about any of that? All right. And then 50% of the material will be on labs 1 through 9 and chapters 1 through 8, missing chapter 7. Any questions about any of that? There will be no trick questions. I personally don't like trick questions. And when I was in college, I had one instructor who just loved giving trick questions. And he had one exam where 50% of the questions were trick questions, and I didn't get any of them correct. Surprisingly, I still managed to be top in the class. So my point is, I don't like trick questions. That doesn't mean I will not ask difficult and hard questions. I'm just not going to ask a question that's tricky. And what I mean by tricky, where you're thinking one thing and you're actually supposed to answer something different. Any questions about any of that? All
So
right.
50% of it's going to be
Chapter 13, 14, and the unknown project.
okay.
Okay.
Thank you.
All right. So obviously, on the unknown project, the best thing to do would be to take a look at the case studies in the lab, meaning take a look at your lab and the case study questions. It will be something like that, where you're given information and then you're supposed to try and identify what you have. And that's sort of what your unknown project is all about. Any questions about that? I may ask you some questions on some tests, but really, I don't think I do, except maybe on the Gram stain, which, if you remember, we had a entire lab as well as a lecture on the Gram stain. Maybe chapter 3 and lab 4. I had more than just the Gram stain, but I'm just saying that I don't think I give any questions on the tests that you may run in the unknown project. For one reason, some of you are going to run totally different tests than other people. All right, so let's go ahead and review chapter 14. I'm going to start from the back and work up because obviously chapter 13 and 14 are the exam. But you'll have the most questions from chapter 13 and 14. The next most will probably be chapter 8 and chapter 1. Chapter 1 is unique in that it introduces a whole lot of topics, and then we come back and later discuss them. And so you can say the exam is from chapter 1, but you could also say it's from another chapter. Like I remember with Cox postulates, we introduced it in chapter 1, but we talked about it at 2 other times in the course. So there will be a question on Cox postulates. There will be a question on the cell wall of bacteria. I don't think there's a cell wall of like archaea or anything like that. And there's nothing about archaea other than maybe the domain. You should know about the domains. Um, and then chapter 1 covers the history of microbiology. And if you do not know, I really do like the history of microbiology. So I tend to ask a few more questions on chapter 1 than all of the other chapters, except for obviously chapter 13 and 14. And maybe chapter 8. Chapter 8, we spent about 2 full days, maybe even a little more than 2 full days covering chapter 8. And microbial genetics is fairly important, so that's why we spent the time on it. So I tend to ask a few more questions on chapter 8 than, say, like chapter 6 or chapter 5. Any questions about any of that? All right, you do need to know the labs, but like I said, there won't be many lab questions on each lab because you've already been quizzed on all of the labs. So there probably will only be something like one or at most two questions from each lab on the final. All right, let's go on to chapter 14. Oh, I already had that open. I didn't close it down from the lecture. Well, you should know the terms of this chapter. As I mentioned, the terms, if you know the terms of this chapter, you'll learn about 50% of the chapter. You should also understand the relationship between the normal microbiota and the host. We're talking about the human host here and the human normal microbiota. You should understand the stages of disease development and understand about nosocomial infections, what they are, and how to control them. You should know where you find the normal microbiota and where you don't find it. You should know about microbial antagonism, and then the relationship between microbes and their host, which is a symbiotic relationship, and it can be commensalism, mutualism, which the normal microbiota are normally that, or parasitism, and then the human pathogens are parasitic symbiotic organisms. All right, you should know about Koch's postulate. This is the third time and last time we talked about Koch's postulate, the different steps, and what you use the Koch's postulates for. We use Koch's postulates to prove that a given microbe causes a given disease. The only exception is if you can't grow the microbe in tissue culture, and if the microbe causes more than one disease, or the one disease is caused by more than one microbe. Like pneumonia is caused by many different species of bacteria, viruses, and probably fungi as well. So you can't use Koch's postulate. There are some exceptions to Koch's postulate. You should know about those. You should know about the difference between a symptom and a sign. You know the difference between a communicable disease and a contagious disease. TB is both communicable and contagious. However, excuse me, TB is communicable, it is not contagious. Measles is both communicable and contagious. You should know the occurrence of disease. You should know about herd immunity. I mean, you should know about latent disease too, but I'm not going to talk about that. Herd immunity is where if enough people in the population are immune, then the disease will not spread to most other individuals. And then the non-vaccinated people are being protected by the vaccinated people. In the United States, we rely on herd immunity because we cannot ever get 100% of the population immunized, which is a problem, and I don't know what to do with it. should know about the predisposing factors to making a disease more likely. You should definitely know about the development of disease. All diseases tend to have an incubation period, a prodromal period, a period of illness, a period of decline, and a period of convalescence. The exceptions are if one of the stages is missing, like the prodromal period sometimes is missing, and then if the disease kills the patient, there may not be a decline or convalescence period. I will not ask you a trick question like which period of illness is non-communicable. I will not ask you a trick question like that. All right, you should know about reservoirs of infection and the three types. You should know about transmission of disease. I will not ask you anything nitpicky about droplet transmission and airborne transmission. I'm not even going to ask you any question about that. Okay, but you should know vehicle transmission and direct contact transmission. And vector transmission. All right, you should especially know about nosocomial infections because this topic is really important to anyone working in the healthcare industry. A disease that the patient gets from checking themselves into a healthcare facility. I didn't mention that because of all the education occurring in the United States and in the hospitals and hospital-like environments, they've actually decreased nosocomial infections in the United States now. So it's closer to 5% than it is to 15%, meaning only about 5% of hospital patients now in the last, 2 or 3 years get a nosocomial infective agent. And that's largely because of the procedures and the education going on in the hospital or hospital-like environment to reduce the spread of nosocomial infective agents. All right, you should know about morbidity and mortality. Any questions about chapter 14? If not, let's move on to chapter 13. Viruses, viroids, and prions. So you should know what a virus is and the different terms to describe it, understand the characteristics of viruses, and the classification of viruses. Remember, with the viral name and classification, all viruses have a common English name. They do not have a Latinized scientific name. You should understand about viral multiplication. That's the replication cycle that the viruses have. And then lastly, understand about prions and viroids. So you should know that all viruses have a genome, which can be either DNA or RNA. It is never both. And then all viruses have a protein coat surrounding that genome. Some viruses have a lipid envelope surrounding the protein coat, but not all viruses do. You should know that viruses display little or no metabolic activity when they're outside of their host cell, and they require their host cell in order to multiply. They need their host cell to get many things like enzymes, nucleic acid, amino acids, ATP, etc. You should know the host range of a virus and what determines that. It is molecules. Let's see if I can blow this up. Molecules on the virus that are called ligands or adhesions that bind to specific molecules on the host cell, which are called receptors. most viruses, not all viruses, but most viruses infect only a few cells of one or a few species. Like HIV only infects a few cells of humans, and then one of the chimpanzees. If you don't know, there are two species of chimpanzee, and only one of them can be infected by HIV. There are some exceptions, like Eastern equine encephalitis. It is a virus that can infect many different species, like almost all mammals. And I don't remember if that one hits insects or not, but there are some viruses that can uh, survive and grow in insects as well. And then it can also hit birds. A lot of those multi-species viruses tend to hit many mammals and many birds. Any question about that? An example would be bird flu, which can spread among almost all birds, and it can spread among many mammals. It can infect humans. The good thing is that bird flu, when it hits humans, it does not readily spread. It can spread, but it doesn't readily spread. I think bird flu actually follows the fecal to oral route, which is why the flu doesn't spread very easily among humans. You don't need to know that. I'm just telling you about host range. And let's move on. You should know the different sizes of viruses, and that is they're much smaller than a prokaryote, about 1/10 the size. And obviously, the largest virus is approaching the smallest prokaryote cell, or actually is a little beyond the smallest prokaryote cell. And then the prokaryote cell is much smaller than a eukaryote cell. Any question about any of that? You don't need to know the exact numbers. You should know the nucleic acid of a virus can be either DNA or RNA, never both, and it can be either double-stranded or single-stranded. Double-stranded DNA is our normal host DNA. Single-stranded DNA is found in our cells whenever our DNA is replicating or being transcribed. It is single-stranded. Double-stranded RNA, that genome is not, or that combination of RNA is not found in our cells, so that's unusual. And then single-stranded RNA is normal in our cells. When we're talking about a viral genome, it's important to discuss whether that single-stranded RNA is positive single-stranded RNA or negative single-strand RNA. And the positive single-strand RNA can be read by the ribosome. So all messenger RNA, for example, of our cells, is going to be a positive single-strand RNA. And then some viral genomes are the complement to that, and that's called the negative single-strand RNA. And I do not know if human cells have negative single-strand RNA in them. If they do, it's definitely not messenger RNA, but I don't know if like tRNA is that, or ribosomal RNA is that, or any of the other RNA in our cell, which I didn't talk about. There are other RNA in our cell. You know about ribozymes, for example. That is a different category of RNA, and it's not a tRNA, messenger RNA, or rRNA. Although that's not entirely true, because there's definitely a ribosome, which is ribozyme, which is definitely rRNA. Anyways, and the viral nucleic acid can be linear or circular. Linear genomes are found in bacteria. Excuse me, linear genomes are found in eukaryotes. Circular genomes are found in bacteria. All right, you should know about capsid and capsomeres. And if there's no envelope, we call it a naked virus. If there's an envelope, we call it an enveloped or an enveloped virus. You know the general morphology. We categorize viruses into a helicovirus, a polyhedral virus, and then the third category is a catch-all complex virus, which can be any shape. I suppose you should know that bacteriophage T4 is the virus that has been studied the most and is understood the best of all viruses. And it's a virus that infects E. coli. All right, viral taxonomy. You should know that viruses are mainly grouped into families. They can be grouped into above that, like an order, but not much above an order, other than the kingdom. We'll talk about that in a minute. And then the family name ends in viridae, and then group below the family is a genus. The genus name ends in virus. Below that is the virus species. And then below that is a subspecies. A subspecies for human would be the different races of humans. For viruses, the subspecies is important because it can give a different, I don't know, disease or category. The best example is the polio virus, where one species causes the polio disease, and then the other subspecies we use as a vaccine to prevent getting the polio disease. Any question about any of that? We don't group viruses much higher than an order because we do not have enough of the genome to understand the evolutionary history of the virus. So we just can't group it higher, other than the kingdom that it infects. And we don't get that from sequencing the genome of the virus. We get it for the cells that the virus infects. And so we do group the virus into a kingdom and then really only into the order, the family, the genus, the species, and the subspecies. Any question about that? It's just that the viruses do not have enough of a genome for us to sequence it and then understand the evolutionary history of viruses. And then they evolve very quickly to confirm or mold to the species that they've infected. So an example of viral taxonomy, the family is the herpes virus, the family would be Herpes viridae. The genus name, herpes simplex virus. The subspecies would be herpes simplex type 1. Yeah. Any question about that? All right. Viruses multiply, and we call this a viral life cycle. For viruses that infect bacteria, meaning bacteriophages, they only have two life cycles, the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, you don't see, well, there it is. There, here it is shown here. The virus infects the host cell, replicates, making new viral genome, and then new viral protein coats and any other protein that's needed by the virus. And then the virus lyses the cell, which will release the newly replicated viruses, which can then go out and infect another cell. And that's the lytic life cycle. A lysogenic virus, once it infects a cell, it has to immediately determine, will it enter the lytic life cycle, in which case it'll go through the life cycle I just discussed, or will it enter the lysogenic life cycle? If it enters the lysogenic life cycle, the virus and the host live in symbiosis, meaning they live together, for as long as the virus remains in the lysogenic life cycle. In the lysogenic life cycle, the viral genome actually integrates into the host cell's chromosome. And that we call a prophage for bacteriophages. And then this can continue for several generations or divisions, where the only time the virus DNA replicates is when the host replicates its chromosome, its DNA. And then the cell will divide. Both of them will have a viral prophage in it. For reasons that are not well understood, every once in a while, the virus can leave the chromosome and then enter the lytic life cycle. Any questions about any of that? All right. Some things to know: in the lysogenic virus, there is a repressor protein in the lysogenic life cycle that when the repressor protein works, it shuts down the lytic genes of the virus. As long as that repressor protein is working, the lysogenic life cycle will continue. Some consequences of lysogeny. The main, you can know about that the cell is immune to infection by that same virus, but the main one is that there is new DNA added to this cell containing the lysogenic virus. And that DNA can give the cell new properties. The best example is Corynebacterium diphtherium, producing the Corynebacterium diphtherium toxin only when the bacteria is infected by the lysogenic virus, meaning it's the lysogenic virus that has the gene for the Corynebacterium diphtherium toxin. Any questions about any of that? All right, that's it for bacteriophages. Let's talk about animal viruses. They do tend to follow the same, life cycles as a bacteria virus. They do have a different release. They can be released by the lytic life cycle, meaning there is animal viruses that follow the lytic life cycle, but they also have a release by budding or a process similar to budding, which I probably don't ask you any questions about, so I won't talk further about it. And then one other difference that animal viruses have that's pretty important is an animal virus can replicate in the cytoplasm, which is where bacteria viruses all replicate in, but an animal virus can also replicate in the nucleus. And a rule of thumb, if the animal virus is a DNA virus, it replicates in the nucleus. If the virus is an RNA virus, the rule of thumb is it replicates in the cytoplasm. There is one exception, and that is the retroviruses are an RNA virus, and they do not replicate in the cytoplasm. And that's the only exception that I know of. Any question about any of that? I guess I should mention that infecting an animal cell is easier because it doesn't have a cell wall, and the infection by the virus happens either by endocytosis or by fusion of the envelope of the virus with the cell membrane. And remember, an envelope is a lipid bilayer, very similar to the cell membrane. And so the envelope can just fuse with the cell membrane, and then that will bring the virus into the cell. All right, I already mentioned that animal viruses can replicate by the lytic life cycle, where they infect an animal cell, replicate, and then lyse the cell, releasing the viruses. So there are animal lytic life cycle viruses, but there are other animal viruses that follow the budding life cycle. And if the animal follows the budding life cycle, it will be an enveloped virus. So the lytic animal viruses tend to be naked viruses, and the budding animal viruses tend to be enveloped. What a budding virus does when it replicates at low numbers. It will push on a membrane. This membrane can be either the cell membrane, the nuclear membrane, or the membrane of the endoplasmic reticulum. And the virus will come to the region of the membrane that has viral proteins in it, and then push it out, making the bud, and then ripping off the bud. And then that bud will reseal, forming the virus envelope. And if this is the cell membrane will reseal. And so the budding virus tends not to kill the cell. If the host cell is killed, it's because the host immune system recognizes this cell as being infected, and then the host immune system would kill that cell. The virus, the budding virus, does not tend to kill the host cell. Any question about any of that? All right, you don't need to know the different families of DNA viruses, but they're shown here. Let me blow that up. And then some of the diseases they cause. You should know the Pox Viridae family encompasses smallpox, and I guess cowpox, which is used as a vaccine for smallpox. You should know a little bit about herpes viridae because we did discuss that one, causing some of the herpes diseases like herpes simplex type 1, herpes simplex type 2, cytomegalovirus, roseola virus, chickenpox. You should know Adenoviridae is a family of viruses that, among other things, cause the human cold. They are the least most numerous family that causes human colds, meaning they're the third family that causes human colds. The other viruses you don't need to know anything about. So DNA viruses replicate in the nucleus. Once the DNA virus enters the host nucleus, it can form a provirus, and if it does that, that provirus will remain in the host DNA permanently for as long as that cell survives. And it will replicate, obviously, if the cell divides. The other thing the virus can do is, once it enters the cell, is produce new virions, making new viral genome, new viral capsid proteins, making new progeny virions, which will either bud out of the cell or they will lyse the cell. And this is a replication of the multiplication of a DNA virus in more detail. RNA viruses, there's more of them. Here's the different families of RNA viruses. You don't need to know most of the family. You might know the Picornaviridae. It's a very large family of viruses, which are very different from each other. Let me see if I can blow that up so you can see it better. This is the family of virus that cause the most human colds. And we call those usually the rhinoviruses. Hepatitis A is in this family, and then poliovirus is in this family. Just looking to see, we've talked about them. You should know a little bit about coronaviridae because of COVID-19. Retroviridae, this is the one RNA family of viruses, which is RNA but does not replicate in the nucleus, in the cytoplasm. This virus replicates in the nucleus. And the reason is it has a DNA intermediate. You probably should know about the Paramyxoviridae, but I'm not going to ask any questions about it. You should know about it because that causes a lot of very common childhood illnesses. But like I said, I'm not going to ask any questions on it. Orthomyxoviridae, you should know about that virus. This is the virus, family of viruses that cause the flu. I mean, I think that's it. All right, any questions about the RNA viruses? Obviously, the replication of the virus will depend on what the viral genome is. DNA replication happens normally. One strand will be used to make the other strand. And that's actually the way double-strand RNA replicates as well. One strand will be made from DNA, transcribed to make messenger RNA, which will be a positive strand, and it can be read by the ribosome to be translated into protein. If we have a single strand RNA, the negative strand RNA makes the positive strand RNA, meaning its template, and then uses that as the template to make the negative strand genomic RNA. And only the positive strand can be translated into protein. And then the positive strand, how it replicates is it makes the negative strand and then uses it as a template to make more of the positive genomic strand RNA. Any question about any of that? And then this is looking at the replication of a positive sense strand RNA virus multiplication in more detail. You should sort of understand that slide. And then the negative antisense strand of RNA virus multiplication in this in more detail. All right, you should know about retroviruses. And they have the enzyme reverse transcriptase, which makes single strand DNA from RNA. And then the reverse transcriptase makes the single strand DNA double strand DNA. The double strand DNA can then integrate into the host cell's DNA. And once that happens, the DNA of the virus will be a permanent fixture of that cell. For as long as that cell is alive, the cell will have the retrovirus in it. And that is actually one reason why when they think an HIV patient has thrown off the virus, they can't say with 100% accuracy because they could have HIV integrated into one of their cells, and then at sometime later in the lifespan, the virus could come out of the genome and start replicating again. Any question about that? See if I can blow this up. So we have positive single-strand RNA, reverse transcriptase in 2 steps, makes double-strand DNA. First it makes single-strand DNA, and then it makes double-strand DNA. That can be transcribed into making messenger RNA, viral messenger RNA, which will make the viral proteins. And the viral protein will then get together with the genomic RNA to make the new progeny virions. And that will bud out of the cell, shown here. Any question about retroviruses? All right, you should know a little bit about coronaviruses because we're still under a COVID-19 epidemic or pandemic, I guess I should say, and that will continue for like 10 or 20 years before they'll say, okay, COVID-19 has become a permanent part of the world human population, and they'll stop it, calling it a pandemic. But that won't happen anytime soon. COVID-19 is likely to continue. Anyways, you should know the three types of coronavirus, actually four types of coronavirus: SARS, MERS, and COVID-19. And then the fourth type is actually the most common type of coronavirus. And this is a virus that can cause the human cold. It is the second most common cause of human colds. And that's actually a coronavirus that causes the human cold. You should know that COVID-19 came about from recombining, not once, but twice, with two bat coronaviruses and a pangolin coronavirus, making COVID-19. And that recombination of COVID-19 has actually happened once again with COVID-19 Omicron. If you remember that virus recombined with COVID-19, recombining with a coronavirus that causes human colds. And that's why Omicron was the least lethal of all the COVID-19 up to that point in time. It was about 50% as lethal as the previous coronaviruses. Although the Delta coronavirus was the most lethal, which is the one right before Omicron. You don't need to know any of that. My point is that coronaviruses do recombine, and that's probably how COVID-19 came about and then first hit the human population. You should know how it's primarily spread by coughs or sneezing or singing from within 6 feet of the patient. It can be transmitted just from speaking or just from breathing, but it's much less likely than from coughing, sneezing, or singing. COVID-19 is likely breathed in, but it can rarely be picked up on your hands, and then you can inoculate your mucous membrane in your face. All right. You should know about viruses and cancer. Some cancers seem to at least get started by a virus, and that's about 10 to 15% of human cancers are started or caused by a virus. I'm saying started because to get a human cancer, you actually need 5 or 6 different mutations, and the virus only gives the first. Human papillomavirus causes cervical and skin cancer. Hepatitis B virus and hepatitis C virus cause liver cancer. You should know that there are 2 ways that viruses can cause cancer. The virus can integrate into the host cell's DNA, mucking up the DNA. Could cause a direct mutation or could just turn off a tumor suppressor gene. I know we haven't talked much about that. Or it could turn on a gene mucking up the DNA. And then the virus can also bring in an oncogene, that is an oncogenic virus. An oncogene is a mutated normal cell gene involved in cellular growth. And the oncogene causes that cell to just grow abnormally. Once again, starting the cancer. Usually, even with the oncogene, you still need, I guess, 4 or 5 other mutations after the oncogene to get the cancer. But my point is the oncogene starts the process. And what it is just a mutated normal cellular gene causing the cell to grow uncontrollably. And the virus brings it into the cell. Any question about any of that? All right. You should know that there is one other life cycle besides the budding in the lysogenic life cycle. For animal viruses, it's a latent life cycle. This is usually a budding virus, which stops causing disease, enters latency, stops replicating, and while it's in the latent stage, the virus is just sitting around waiting until it leaves the latent life cycle, re-enters the budding life cycle, and then causes disease. And when it's in the budding life cycle, it's replicating. Any question about any of that? The best examples of a latent virus disease is herpes simplex type 1, which is in the budding life cycle when you have the cold sore. And when you do not have the cold sore, the virus is in the latent life cycle. The other example is chickenpox, which is in the budding life cycle when the child has chickenpox. And then the virus enters latency. It may remain latent for decades. And then it will re-enter the budding life cycle, causing shingles. And that's actually one reason to get the chickenpox vaccine. And that is if you never get chickenpox, you can never get shingles. Any question about any of that? I won't ask you a question about the chickenpox vaccine. Oops, let's talk about this. Here's an acute viral infection. The virus infects the patient very quickly, replicates, causing high number of infective particles, causing disease, and then the virus quickly goes away. Human cold would be a good example of an acute viral infection. There's also a persistent viral infection where the virus starts at a very low number and over time increases, starting with a very mild disease. Over time, the disease gets worse. And you then have the bloom, where if the virus kills the patient, obviously the chain will be broken. Not all persistent infections kill the patient. Some of the persistent infections will go to this and then plateau. Oops, sorry. My mother had shingles, which entered the persistent infection, where she always had shingles for the last few years of her life. And what happened was it was a persistent viral infection and just plateaued. Trying to think of another example, I just can't think of it. Cytomegalovirus. Some people have a cytomegalovirus infection, and over time, their symptoms will get worse, and then the patient starts tolerating it, and the viral infection just plateaus. Any question about that? HIV followed the persistent infection in the old days, where the patient would get worse over time, and then it would bloom, causing AIDS, and then the AIDS would kill the patient in the old days. A latent virus infection like chickenpox, this would be chickenpox when the child gets chickenpox, the virus is then gone. It's latent. You don't even see it in the patient. You could find it in the patient if you were to actually do, I don't know, what do you call it? Synthesize the patient's DNA and then look for the chickenpox virus. But that would be the only way, and it'd be very low numbers. It's not replicating. And then it can come out. They call that a latent infection, and the virus starts budding, and the patient gets starts releasing viruses, and will have the shingles for the chickenpox disease. Any questions about any of that? All right, you should know about prions. They are proteinaceous infectious particles. The proteins reproduce because the cell has a normal protein, PRP superscript C, which the nerve cell needs, and then the prion protein, PRP superscript SC for scabies, which the prion protein comes up to the normal protein, causes the protein to change its folding pattern into the prion protein. Now the cell has two prion proteins, and it'll be short on the normal protein, so the cell will make more of the normal protein, and then each of these will come up to the normal protein, making more of the prion protein, so that the prion protein accumulates in the nerve cell, forming plaques, and eventually will kill the cell, causing prion disease. Any question about any of that? I don't think I ask you anything about the brain, but it's called spongiform encephalopathy because the brain of the prion patients or the animal looks like a sponge compared to the normal brain. All right, you should know about plant viruses and viroids. Plant viruses cause diseases that are economically important to people, but they only cause diseases on plants. And so we're not going to talk any further about them. And then prions also cause diseases, excuse me, not prions, viroids also cause diseases in plants. They are a naked RNA molecule, which is infectious. They only cause diseases in plants, though. There are no known viroids affecting animal cells. Any question about any of that? All right, that's it for chapter 13. Let's move on to chapter 8. Well, you should know about what a genome is, gene, genotype, phenotype, vertical, horizontal gene transfer, what an operon is, what a promoter is, an operator, what a structural gene is. You should know about crossing over, transformation, conjugation, and transduction. You should also be able to describe DNA replication, RNA transcription, and protein translation. And then lastly, you should understand about the different types of mutation. So you should know these terms. You should know that DNA is made up of only 4 nucleotides, and you can learn that either as adenine, thymine, cytosine, or guanine, or A, T, C, and G. DNA in our cells is normally double-stranded. One single strand, is held together by a sugar phosphate, that's a sugar phosphate backbone, and this is a covalent chemical bond holding the single strand together. The double-strand DNA is running in opposite directions. We call that the strands are anti-parallel. This is running 5' to 3' this way. This one is running 5' to 3' this way. The double-strand DNA is only held together by hydrogen bonds between the bases of the single-strand DNA. Any question about any of that? Hydrogen bonds, there's 2 of them between A and T, and 3 between C and G. You know the vertical flow of information in a cell. Normally information moves from DNA to RNA in transcription, and then from RNA to protein in translation. The only exception of this, I'll mention that now, is in retroviruses. where reverse transcriptase can take information in RNA and make DNA. And then from the DNA it goes in the normal flow. All right, you should know about the flow of information within a cell, between generations of cell, or in humans, we could say from two parents to their offspring, and then from one cell to a cell of the same generation, the recombinant cell. Normally, the DNA leaves one cell and then would be taken up by the recombinant cell. Any question about any of that? All right, you should know about DNA replication and RNA transcription. I'm not going to go into the details, but you should know it. Here's something we should mention. The genetic code has about 64 different codons, but organisms, at least humans, only have 20 different amino acids. So there is some redundancy of the genetic code. See if I can blow this up. Meaning there is, for most of the codons, most of the amino acids, there's more than one codon to make that amino acid. Phenylalanine, for example, there's two possible amino acids to make the same amino acid, meaning two possible codons to make the same amino acid. For leucine, there's actually 6. For serine, there's 4. There's only 2, which there's only one amino acid, one codon to make that amino acid. And that happens to be tryptophan and methionine, at least for humans. So you should know about protein translation. All right, you should know about the one way to control bacteria contents is to control the gene expression. We're going to look at regulation pre-transcriptional control by an inducible operon, and we're going to look at the lac operon. Eukaryotes have similar operons. The only difference is that bacteria operons may have more than one structural gene in the operon. In a eukaryote operon, there is only one structural gene in each operon. And for the lac operon, for example, in bacteria, we have 3 structural genes, which are all related to lactose metabolism. We have the 3 same genes in eukaryote cells. It's just that there are 3 operons instead of 1. Any question about any of that? And then eukaryote operons have more regulation than a bacterial operon. A structural gene is the gene of interest. So normally when we're talking about a gene, we mean a structural gene. An operon will involve a structural gene, perhaps more than one structural gene if it's a bacteria, and then the controlling regions around that structural gene, specifically the promoter, the operator, and the terminator, which is not shown here. The promoter we've already discussed, and that is the region of DNA where the polymerase binds to the DNA. And this will be an RNA polymerase because it's transcribing the gene. The operator is where the regulatory gene binds to the operator, stopping the promoter from transcribing the gene. The regulator is made by another gene, which in the case of the lac operon happens to be nearby. In most cases, like in a eukaryote, this gene is nowhere near the gene it's regulating. In fact, in eukaryotes, it's usually on a different chromosome. And that is how the lock operon is shut down, is that the repressor protein binds to the operator, shutting down gene transcription. You need a way for the lac operon to be turned on. And what happens is we have an inducer, which is allolactose. Allolactose is around whenever the sugar lactose is around. They are isomers of each other. One is a left-handed and one is the right-handed. Allolactose can bind to the repressor protein, inactivating it, which will eventually turn on the lac operon. Any question about any of that? In the case of bacteria, we have an unusual messenger RNA which is made, and this messenger RNA encodes 3 proteins. Usually we only have one. At least in eukaryotes, you only have one. We've only talked about one, but in this case, from this messenger RNA will be made the lactase enzyme called beta-galactosidase, the permease transport protein, which brings the sugar lactose into the cell, across the cell membrane, and then transacetylase, which is not discussed. It's a protein which helps convert beta-galactose to the sugar glucose for metabolizing lactose. Any question about any of that? All right, you should know about mutations. You can have a mutagen, which can cause a mutation, or a spontaneous mutation, which is usually happens just normally on its own. Whenever the DNA is replicating, if it makes a mistake, we call it a spontaneous mutation. It doesn't happen very often, but it does happen. You should know about the different types of mutation. A base substitution, which is when a single nucleotide base is changed. A frame shift mutation, where a nucleotide is either added or deleted, or it could be a multiple of several nucleotides, as long as it's not a multiple of 3. We call that a frame shift mutation. And then there's a macro mutation, which is when an entire region of a chromosome is either added or deleted. And remember, a macro mutation is not discussed in the textbook. A silent mutation is where the DNA nucleotide is changed, but it does not change the amino acid, like when UUU is changed to UUC, it still codes for the phenylalanine. That is called a silent or a neutral mutation. No effect on protein function. A missense mutation is when we change the nucleotide of the codon. So instead of GGC coding for glycine, we have it changed to AGC, changing the nucleotide, which also changes the amino acid. A missense mutation, where one amino acid is changed. And that's a missense mutation. A nonsense mutation is where you change one nucleotide in the codon, and instead of coding for an amino acid, it codes for a stop codon. That way you will get a truncated protein, a protein smaller than the original protein, a nonsense mutation. A frame shift mutation, the easiest way is to add or remove a base. When you remove a base, what used to be U will now go UU, and then it'll grab this G from the next codon to go UUG, changing this codon, but then it changes all later codons because the frame shift has been changed. And that could potentially change all of the amino acids after this frame shift. Any question about any of that? The only exception is if you remove or add 3 nucleotides or a multiple of 3. If you were to add, well, let's say remove this one codon, UUU, that's the only thing that would be removed. Instead of having methionine, lysine, phenylalanine, and then glycine, you'll have methionine, lysine, and then glycine. The thing is that the frame shift is not changed. You've only changed that one amino acid. So what would that be? That would be like a missense mutation, because only one amino acid was changed. All right, 2 causes mutation, spontaneous and mutagens. You should know ionizing radiation is radiation with a lot of energy. When it hits the atom or the electron, it causes the electron to leave the atom or the molecule, causing an ion, which can then bump into DNA and damage the DNA. UV radiation is a form of non-ionizing radiation. What it does is cause thymine dimers to adjacent thymines, can then actually pair with each other, which doesn't normally happen, and then they will not hydrogen bond with the other strand of DNA, and that will cause a bubble in the DNA. This DNA cannot be replicated, at least normally, and cannot be transcribed normally either. There's also a chemical mutagen. Nitrous acid was the example where the nitrous acid comes up to the adenine and then makes this adenine not act as an adenine, and it acts like a guanine, and then the polymerase will read that as a guanine and then add a C there, and then that when it further not replicated, that will permanently change the genome of this cell. All right, you should know about genetic recombination, the exchange of genes between 2 molecules of DNA. That often happens with crossing over, which normally happens in reproducing egg or sperm cells. Is that right? Yes. Where crossing over happens, the mother's chromosome gets together, pairs with the father's chromosome, and then they cross over, recombining. Recombining is more general, where the exchange of genes may happen between pieces of DNA. Doesn't have to be crossing over. Recombination is important for populations because it can allow new genetic combinations, such as the blue-eyed gene being paired with the red hair gene. Assuming those weren't together. I mean, they may have been together when they first came up, but I don't know. My point is that it can allow new genetic combinations to occur in a population. Vertical gene transfer happens during reproduction between generations of cells. Horizontal gene transfer happens when genes or DNA move between cells of the same generation. So the DNA leaves the donor cell and then enters the recipient cell. Horizontal gene transfer can happen by transformation, conjugation, and transduction. You should know transformation is when the cell takes up DNA, expresses it, which can change the phenotype of that cell. Transconjugation can happen in 2 ways, depending on a gram-negative cell or a gram-positive cell. Gram-negative cells, the F plus cell here, makes a sex pilus, which is hollow. It skewers this cell and then can allow a plasmid to leave this cell and then enter that cell. The plasmid replicates, it doesn't leave the cell. It sends a copy into that cell. And then the mating bridge can also happen with conjugation. Transduction is when a virus moves DNA from one cell to another. So the virus inserts itself into the cell, degrades those chromosomes, replicates itself, and a mistake gets made where this virus, instead of getting viral genome, gets the piece of DNA from this cell. The virus can then add the DNA from this cell to this cell, and that DNA can then integrate into the host chromosome and then be expressed. And that's how transduction moves DNA from this cell to that cell. Any question about any of that? All right, you should know about plasmids. They're a small molecule of DNA in a cell, usually only in bacteria, archaea, or yeast cells have plasmids. They only have a few genes that differ from the chromosome, which can have thousands or hundreds of thousands of genes. So it's a much larger molecule. The plasmids are important because plasmids are more likely to spread between cells than chromosomes. Let me go back here. So you're more likely to get a plasmid. Obviously, you have to have a plasmid. Well, no, you can't. You're more likely to have a plasmid happening by conjugation or by transformation. I guess not by transduction, but by transformation or conjugation, more likely to happen with a plasmid than a chromosome. And transposons are pieces of DNA that move from one region of the DNA to another region of the DNA. And that's important because it can put new genes together. And then it can also move a gene from the chromosome to a plasmid, and then plasmids are more likely to spread between cells of the same generation of cells. Any question about any of that? All right, so you should know about genes and evolution also. Any question about chapter 8? I didn't mean to spend so much time on chapter 8. Let's see if I can speed this up. Chapter 6, so you should know the different terms. What's a psychrophile, a psychotroph, a mesophile, thermophile, hypothermophile, acetophile, halophile, aerotolerant anaerobe, facultative anaerobe. You know what binary fusion is, reproductive spores, and endospores. And know the difference between an endospore, which is a cell survival technique, and a reproductive spore, which is a reproductive technique, the way some cells reproduce. Most cells that are bacteria reproduce by binary fission, but some bacteria can reproduce by budding, and some can reproduce by reproductive spores. Understand the various requirements for growth, and then the 4 phases of growth, and then the methods we have for measuring microbial growth. Know that you have requirements for microbial growth, both physical and chemical requirements. So you know the temperature requirements, the different names, their pH requirement. Acidophiles are something that wants to grow in an acid environment. Osmotic pressure, so the salinity requirement, or maybe sugar requirement. Halophiles, something that likes growing with salt, a high salt concentration. You know that all organisms require trace elements Usually they just get their trace elements from their environment. We get our trace elements simply from the food we eat and the liquids we drink. If an organism requires an organic growth factor, that they must get. An example of an organic growth factor for humans would be vitamin C. We must get it in our diet. Cells have a requirement for the oxygen concentration, and that's because there are 4 toxic forms of oxygen: singlet oxygen, superoxide, free radical, peroxide anions, and then hydroxyl radical. All of these are toxic, and the cell must remove these 4 toxic forms of oxygen, or the cell will die if it is exposed to oxygen. And that includes our cells too. You can look at the microbe's requirements for oxygen by putting the microbe in a thioglycolate media. And if the growth is mainly at the top, it's an obligate anaerobe. If the growth is mainly at the bottom, it's an obligate, did I say aerobe? Obligate aerobe. Here is an obligate anaerobe. I might have said that wrong. If the growth is throughout the tube, but mainly at the top, it's a facultative anaerobe. If the growth is uniform throughout the tube, it's an aerotolerant anaerobe. There are only a few bacteria that I know of that are this way. And then there are some bacteria that are microaerophiles. Elobacter pylori, a bacteria that grows in our stomach, is a microaerophile. It requires oxygen, but it prefers having less oxygen than 20%. Any question about any of that? Should know about media, agar, different types of media, complex media, anaerobic media, selective media, differential media, selective and differential media, and then an enrichment media. Remember, an enrichment media is selective, but we use it to enrich for the occurrence of cells of interest. You know about how to streak a plate for colony isolation, and that if you want to get a pure culture, you need to streak the plate out 2 times, getting the cells through 2 colony formation events. And then it's not guaranteed it'll be pure, but at least it'll be 95% or higher chance that it'll be a pure culture. Already talked about that. All right, the phases of growth. You should understand this bacterial growth curve. Cells normally enter a lag phase where they're making the enzymes they need to grow in the new environment. The cells enter the log phase. We then enter the stationary phase because the cells are having cells equal number growing and equal number dying. The lucky cells have some nutrients around, not too many waste products around, and some space around them. The unlucky cells have lots of waste products around them, very little space around them, and not very many nutrients around. And so the population number is about the same, stabilized, because the number of cells growing equals the number dying. And then you enter the death or decline phase, where the number of dying cells outnumbers the number of reproducing cells. And late in the death or decline phase, you have no reproducing cells. You know the different methods we have for counting microbial growth. All right, any questions about this chapter? Let's move on to chapter 5. You should know the terms of this chapter. You should understand what feedback inhibition is and understand what aerobic respiration is. We went into great detail about what aerobic respiration is, its steps, and how it differs from anaerobic respiration. You know, microbial metabolism is separated into two parts, catabolism and anabolism. You know about metabolic pathways. You should know about the collision theory and how chemical reactions occur when atoms, ions, and molecules collide with each other. Shown here, an enzyme reduces the activation of energy compared to the activation energy without the enzyme. If this is, well, I guess glucose in our cells, making CO2 and water, this happens in our cells at a temperature of 37 degrees. You can have this same reaction happening outside of cells without an enzyme, but you have to take a blowtorch to the glucose to burn it to CO2 and water. And that would be raising it to like about 1,000 degrees. I don't know if that's Fahrenheit or Celsius. It's one of those. It's quite a bit higher than 37 degrees Celsius. Any question about any of that? So you should know what enzymes are, what they do. You know, enzymes don't work in a vacuum. They may need a coenzyme or a cofactor. You should know two of the important coenzymes, NAD plus and FAD. You should know how enzymes work. Here's the lock and the key model, where the enzyme binds to the substrate, like a key binds to a lock, and then it puts stress on the molecules, what do you call it, the atomic arrangement, the chemical bonds of the molecule to make the product. And then the enzyme falls off and then can recombine with another substrate. Here's the hand in the glove model, where the substrate binds to the glove, I guess, and then the glove actually molds to the shape of the hand, changing its shape. And I prefer the shapeshifter's hand going in the glove, where the glove molds to the shapeshifter's hand, but the shapeshifter's hand actually molds to the glove as well, meaning changes its shape to mold the enzyme. And that's really the way enzyme and substrates work. As I mentioned, enzymes don't work in a vacuum. For their optimal working, they have to be at the correct temperature, the correct pH, the correct salinity. Generally, heavy metals have to be absent. Alcohol has to be absent. UV radiation has to be absent. All of these can cause the protein to denature. Enzymes do need to have a substrate around. If there's no substrate around, the enzyme can't act. The enzymes don't want poisons around. The poisons can destroy the enzyme. You can also have inhibition of the enzyme, such as competitive inhibition or non-competitive inhibition. And then there's also feedback inhibition. I'll talk about those in a minute. There's an enzyme, and there's a denatured enzyme, taking on a different three-dimensional shape. Just showing you how temperature and pH can affect the activity of an enzyme. This enzyme works best around 37 degrees. When you move away, the enzyme decreases its activity until you move way away from the temperature. The enzyme won't have any activity. The effect of substrate concentration on an enzyme activity. Competitive inhibitor is where the inhibitor competes with the active site it competes with the substrate for the active site. And that's competitive inhibition. Non-competitive inhibition or allosteric inhibition is where the non-competitive inhibitor or the allosteric inhibitor binds at a site other than the active site, changing the three-dimensional shape of the enzyme So the enzyme can no longer bind to the substrate. And then here's feedback inhibition, where we frequently see this in a cell where the end product of the reaction actually binds to the first enzyme, inactivating the enzyme, making no more intermediate A or no more intermediate B, no more end product. And then when the end product decreases in the cell, it will fall off the enzyme. And then the first enzyme can be active and make more of the end product. That's feedback inhibition. Any question about feedback inhibition? You should know about ribozymes, RNA enzymes. You should know about metabolic diversity among organisms. Let's break it down into this slide. Let's go down here. Photoautotroph is an organism that gets its energy source from light and its carbon source from CO2. Green plants, everybody knows that's a photoautotroph. An animal is a chemoheterotroph. It gets its energy need from glucose or some organic food, and then its carbon source from some organic compound such as glucose. You can have an energy source being an inorganic chemical, and that would probably be a chemosynthetic bacteria. All the animals are chemoheterotrophs. There are some chemosynthetic bacteria which are chemoautotrophs. The difference is they get their energy source from a chemical, usually an organic chemical, and they get their carbon source from CO2. And then the photoheterotrophs get their energy need from light, and they get their carbon from an organic chemical compound such as glucose. You don't probably need to know about an example because you probably don't know a green and purple non-sulfur bacteria. Any question about any of that? All right, you should know about oxidation reduction reactions. You should know about aerobic respiration. This slide is showing you an overview of aerobic respiration. You should know that ATP is made in 3 ways. The first way is by substrate level phosphorylation, where phosphate is moved from one molecule, the ADP, to make ATP. ATP can also be made by respiration, which involves the sequential transfer of electrons from an electron donor to an electron acceptor, and in the process, ATP will be made. And then ATP can also be made by photophosphorylation, which only happens in photosynthetic cells where ATP is made, although it is often used up to make sugar. All right, so many organisms can digest carbon and carbohydrate metabolism to make energy. So the most common energy source is glucose. And this can happen both with respiration and fermentation, and know that there's both aerobic respiration and anaerobic respiration. Here we're looking at an overview of aerobic respiration versus fermentation. Both of them have glycolysis in it. Aerobic respiration has glycolysis, the Krebs cycle, and the electron transport chain. Fermentation has glycolysis, and then 1 or 2 fermentation steps. Much higher ATP in aerobic respiration. This is an overview of aerobic respiration in a eukaryote cell. Here would be the cell. Glycolysis happens in the cytoplasm. The pyruvate then moves into the mitochondria, so does the NADH. We'll talk about that later. Pyruvate then enters the Krebs cycle, which some ATP is made in glycolysis, and in the Krebs cycle, most of the Energy, though, from glucose will be shunted by NADH and FADH2 into the third step of aerobic respiration, which happens along the inner mitochondrial wall of eukaryotes, in and across the inner mitochondrial wall. And this we're going to call electron transport chain. So glycolysis, you should know the overview. You get a net gain of 2 ATP, 2 pyruvates, and 2 NADH. The pyruvate then enters the preparatory step and the Krebs cycle, making some ATP, some NADH, some 1 FADH2 per pyruvate, and then 3 CO2 per pyruvate. But remember, you have to multiply this if the question asks per glucose. You multiply everything by 2. And there's the Krebs cycle. And then we enter the electron transport chain, where the NADH and FADH2 carry the electrons and the hydrogen to the electron transport chain. Where do I blow this up? There we go. When the electrons move through the electron transport chain, some of the proteins get the energy they need to pump hydrogen ions from this side of the membrane to that side of the membrane, creating a potential energy difference between this side of the membrane and that side of the membrane. These hydrogen ions will want to move to this side of the membrane. We can only do that through this protein, which is an ATP synthase protein. And when the hydrogen ions move from this side of the membrane to that side of the membrane through this protein, it gives this protein the energy it needs to synthesize ATP from ADP. The other thing going on is the electrons will end up at oxygen, which will convert, combine with the oxygen and the hydrogens to make water. So all of the oxygen you breathe in is converted to water. All of the CO2 you breathe out, which is actually shown in the Krebs cycle, happens from the burning of glucose. Final electron acceptor in aerobic respiration is oxygen. It's the most efficient way of making ATP. In eukaryotes, ATP production happens in the mitochondria, although glycolysis happens in the cytoplasm. In prokaryotes, glycolysis happens in the cytoplasm, and then the electron transport chain happens in the cell membrane. So this right here is the cell membrane of prokaryotes, and right there it's the inner mitochondrial membrane of eukaryotes. Let me back up. So in eukaryotes, we get 36 to 38 ATP produced per molecule of glucose. And obviously you use up 6 oxygen, 38 ATP, 38 phosphate, and you generate 6 CO2 from the burning of glucose. And you generate 6 water from breathing in the oxygen. Prokaryotes are a little more efficient. They always generate 38 ATP. All right, anaerobic respiration is similar, but it has a shorter electron transport chain. Depends on the form of anaerobic respiration, meaning there's many different ones. It could be nitrate ion, it could be nitrite ions, it could be sulfate ions, it could be iron. There's many different forms of anaerobic respiration. The ATP yield is always greater than 2 and up to 36, always less than 38, depending on which form of anaerobic respiration it is. Some are more efficient. The most efficient, though, are not as efficient as aerobic respiration. So therefore, anaerobes tend to grow slower than aerobes. Fermentation, we do generate ATP from glucose fermentation, but you only generate a net gain of 2 ATP. And all of that happens in glycolysis. Fermentation has a fermentation step or two, and the main point of that step is to convert pyruvic acid to the fermentation end product, and then convert NADH made in glycolysis back into NAD+. You need the NAD+ present to allow fermentation to occur. Any question about any of that? You should know the two common forms of fermentation, lactic acid fermentation and alcoholic fermentation. There are other types of fermentation shown here, but I won't discuss them now. All right, you should know that glucose is the preferred molecule for, well, any respiration, aerobic or anaerobic, or even fermentation. Other foods can be used. Carbohydrates will be broken down and converted into a sugar. If it's not glucose, the sugar will be converted into glucose. Lipids and proteins can be broken down as well. It's a little more complicated. Lipids and proteins are too big to enter the cell, but the cell can secrete lipases or proteases outside the cell to digest the cells into either amino acids or what would that be there? Fatty acids and glycerol for the lipids. And then those can be brought into the cell and then digested. Most of the time for us couch potatoes, they will be converted into glucose and then sent through aerobic respiration in the normal way. You can burn lipids directly, like a marathon runner, after running 45 minutes, will start to burn lipids directly. Any question about any of that? All right, you should know about the integration of metabolism. We have amphibolic pathways that can work both ways. And that's why when you make lipids, most of us couch potatoes will convert that lipid into glucose, and then glucose will be sent through aerobic respiration in the normal way. I guess an easy way to state that is the other way, and that is if you eat too much carbohydrate, everybody knows we're not going to store that primarily as a carbohydrate. We'll convert that into a lipid. And that's true if you eat too much protein as well. All right, photosynthesis. Let's talk a little bit about that. You should know there's two types, cyclic and non-cyclic photosynthesis. In cyclic photosynthesis, we make glucose, 6 oxygen and 6 water. You should know this equation. In cyclic photosynthesis, we only make ATP. And photosynthetic cells can generally make either cyclic photosynthesis or non-cyclic photosynthesis, depending on what they need. If they need glucose, they'll undergo non-cyclic photosynthesis. If they only need ATP, they'll engage in cyclic photosynthesis. Cyclic photosynthesis is very similar to aerobic respiration. We have light, I guess that's different, which hits an electron from chlorophyll in a green plant, at least, or a green bacteria, that causes an electron to move to this protein in the membrane, to this protein in the membrane, to this protein in the membrane, back to this protein in the membrane, and then the electron moves back to the chlorophyll. That's why it's called cyclic photosynthesis. The electron just moves in a cycle, returning to where it started. Along the way, the electron, when it's moving through this protein, gives this cell the energy it needs to move hydrogen ion from this side of the cell to this side of the cell. That hydrogen ion gradient can be used to then make ATP. Hydrogen ions will want to move through to this side of the membrane. They can only do it through this protein, which is an ATP synthase. When the hydrogen ion moves through this protein, it gives the protein the energy it needs to make ATP. Any question about any of that? All right. Let's move on to chapter 4. I'm trying to speed this up. All right, you should know the functional anatomy of prokaryotic cells. I do not believe I'm asking you any questions about eukaryotic cells, other than perhaps the difference between prokaryotes and eukaryotes. So you should know the different three main shapes of bacteria. Can be different shapes. You should know that most bacteria are monomorphic, but a few are pleomorphic. You should know the different cellular arrangements that cocci can have, and you should know that cocci have more cellular arrangements than any other cell. Bacteria, I should word it that way. There can be singlets, which isn't shown here except for maybe right there. There can be diplococci, streptococci, tetrads, Sarcenia, and then staphylococci. The bacilli only have singlets shown here, diplobacilli, streptobacilli, and then palisades. The coccobacillus is not a cellular arrangement, that's a shape. Any question about any of that? All right, here's a question for you. What cellular arrangement can spirochetes have? Think about that. All right, you should know the typical structure of prokaryote cells, and then you should know that what's shown in red is found in all bacteria cells: cytoplasm, ribosomes, plasma membrane, and the nucleoid containing the DNA. So that would be the nucleoid region. Remember, in a prokaryote, the nucleoid region is wherever the DNA is floating in the cytoplasm. There is no distinction between the nucleoid region and the cytoplasm because there's no barrier. All cells actually have cytoplasm, ribosomes, plasma membrane, and DNA. You know about the glycocalyx, flagella. You don't need to know the flagella arrangements. You should know that if bacteria move, it's because they have one or more flagellum. You should know about the endoflagella. That's a specific flagella that is found in spirochetes, where the endoflagella is inside the cell wall. Let me show that. There's the cell wall. Blow it up a little. Oh, there's better. So the endoflagella is inside the cell wall, and then it wraps around the cell. causing the spirochete movement. Fimbriae, the sex pili, prokaryote cell wall, peptidoglycan, gram-positive and gram-negative bacteria cells. You should know the difference between them. You should know about the atypical cell walls. Mycoplasma are bacteria, but they lack a cell wall. The archaea are atypical in that they are not bacteria. They're their own domain, and they do not have peptidoglycan. You don't need to know this. They have pseudomurine in their cell walls, or they have no cell wall. And then the Mycobacterium are atypical bacteria that have different cell walls. They have mycolic acid in their cell wall. You know about the plasma membrane and the fluid mosaic model. You know about movement of materials across the membrane, passive and active processes. You know about group translocation. It is a special active transport that only prokaryotes have, found only in prokaryotes. All right, you know about endospores. This is a feature that mainly two families, Bacillus and Clostridium, make. I think there's one or two other families, but there's only like one or two members in those other families. They make an endospore, which is a survival structure. One cell makes one endospore, then the parent cell dies, so one cell makes one cell, and that's why it's a survival structure, not a reproductive structure. The endospore can survive very harsh environmental conditions where the parent cell cannot. All right, I think that's it for this, except for the last three slides. The endosymbiotic theory is a theory to explain how eukaryote cells obtain their mitochondria and their chloroplasts. These organelles are unusual in that, first of all, they're double membrane. They have DNA in them, and they have ribosomes in them. Very different from all other organelles in a eukaryote cell. And the theory is that they came from a bacteria that was taken in by a proto-eukaryote cell. And then the two started living in symbiosis. And now we no longer find the original bacteria, which has become the mitochondria, or the chloroplast. We no longer find them free living. We only find them living in the eukaryote cell. And then the proto-eukaryote has evolved into the eukaryote cell. As you know, the evidence for this theory. All right, any question about any of that? If not, let's move on to chapter 3. You know the terms of this chapter. Observing microorganisms. You know why we use the metric system, mainly because science and medicine use it. And then all the countries in the world, except for these three, use the metric system. And two of these countries are converting to using the metric system. The United States is one, it's just that we're converting very, very slowly. You know the sizes of things. You do not need to know the exact size, but you should know the rank order. And then the parts of the microscope you should know. Difference between a compound light microscope and a simple microscope. A simple microscope has only one magnification lens. A compound microscope has two, the eyepiece and the ocular lens. Not the eyepiece, the eyepiece, which is the ocular lens, and the objective lens, sorry. Should know what resolution is. Refractive index. While oil helps with refraction. Probably don't ask you anything about bright field illumination versus dark field. I might ask you something different between an electron microscope and a light microscope. All right, you should know that staining, all it does is increase the contrast between the background of the slide and the specimen, and these cells are unstained. So all it's doing is increasing the contrast. You should know there's basic staining, which are most of the stains in microbiology, which stains the cell. And then there's negative staining, which stains the background of the cell. Both of them allow you to see the specimens much better than without the stain. You should know there's three different types of stains: a simple stain, a differential stain, and a special stain. A simple stain simply uses a single dye. A differential stain usually uses two different dyes, but that is not required. There are two common differential stains, the Gram stain and the acid-fast stain, used in microbiology. You should know about the Gram stain procedure. I'm not going to go over it now, but you should know that. The basis of the Gram staining. And I will not ask you any questions about the special stains. Staining a special structure in the cell. Any questions about chapter 3? All right, any questions about chapter 2? Chapter 2 is the chemistry of life. And I don't think I have any questions on the final directly from chapter 2. I will expect you to understand what an organic compound is and what molecules are, but I don't believe I have any questions directly on chapter 2. Any questions about chapter 2? No, let's go on to chapter 1. All right, you should know the importance of microbes in the ecosystem as well as to people. You should know a brief history of microbiology. You do not need to know the year of anything in the history of microbiology, with the exception of the first microbes that we have evidence in the fossil record on Earth. Other than that, you don't need to know any of the years in the history of microbiology. What you should know is who discovered what, and obviously what they discovered. And then you should know the terms, importance of microbes in the nitrogen cycle. Wherever there's a red arrow, this step in the nitrogen cycle is happening because of a microbe. Microbes are important in making soil. Microbes were and are today still important in making oxygen on the planet Earth. About 50% of the oxygen we have present on the planet Earth is from microbes. 50% comes from plants. And then microbes were the first ones who started making oxygen, which put oxygen in the atmosphere, creating the ozone layer, which then allowed the evolution of green plants. Microbes are important in the breakdown of matter in a sewage treatment plant. You know what a microbe is and what a cell is, where we find microbes. Do you know what a pathogenic microbe is? You know the beneficial roles that microorganisms play in the planet, as well as beneficial roles that microbes have for humans, like making industrial chemicals, making certain insecticides, fermenting foods. All of our fermented foods happen because of microorganisms. Products made in manufacturing, antibiotics are made by microbes, and then other products like insulin is made by microbes. All right, so know about the scientific names started by Carlos Linnaeus. We still use his naming convention where every species is giving two names, the genus name and then the specific epithet name. So for humans, the species name is Homo sapiens, Homo being the genus name, sapiens being the specific epithet. And this is called the scientific nomenclature, also called the binomial nomenclature, because all species has two names. You should know that all species are Latinized. The name is either italicized or underlined. The genus name is capitalized. The specific epithet is lowercase. The name usually is descriptive or honors a scientist. Like Homo sapiens is descriptive, meaning wise man that walks on 2 feet or 2 legs, one of the two. And then Escherichia coli, coli being descriptive, found in the colon, and then Escherichia honoring Dr. Escher, an early microbiologist. Should know in the classifying of organisms, we break all living things into 3 domains: the domain bacteria, the domain archaea, the domain eukarya. These domains are defined, and all you need to do is sequence the DNA of a species, and you will find it belongs to one of the domains, because each organism in a domain will have a more similar sequence in DNA to any organism outside of that domain. So the domain is defined, and it is defined on the genetic sequence of the organism. We then separate the domains into the kingdoms, and this is in debate how many kingdoms there are and what's the definition of a kingdom, meaning there isn't any. And we will simply use the 6-kingdom approach, where we have the kingdom bacteria, which is the same as the domain, the kingdom archaea, which is the same as the domain, and then the domain eukarya, we split into the plants, the animals, and the fungi. And then lastly, we split the other eukaryotes into the protista. The protista are all eukaryotes that are not a plant, not an animal, not a fungi. And so it is not a monophyletic group. Any questions about any of that? You should know the difference between the domains. And then you should know in the history of microbiology, the major events, who discovered them. You do not need to know the year except for this first one. Any questions about chapter 1? I guess I should mention that the early years of microbiology were largely trying to answer the question, and that is, did the theory of spontaneous generation give rise to microbes, or did the theory of biogenesis give rise to microbes? And Louis Pasteur finally disproved the theory of spontaneous generation, thereby confirming the theory of biogenesis, and that is microbes come from pre-existing microbes. You should know how he did that too. All All right, you should know the definition of an emerging infectious disease, and that's all you need to know about an emerging infectious disease. Any questions about chapter 1? All right, if there's no questions, that's it. Good luck on your final. If you have any questions, please email me. I'll try and check my email every day until at least the final.
I have a question.
Uh-huh.
What is final dash A? It's 15 points. Is it like a questionnaire?
It is a short answer final questions. There's only a few questions. I don't remember, it's 3 or 5, something like that. But it is short answer questions where you might be asked a question like from the unknown project where you're given certain information and you're trying to identify the species. And obviously, I'll give you the different tests that were run and a key. So you might be given something like that. So this will take time, and that's why it's in the short answer. I might ask for a sentence. I don't think I have any paragraph answers, but it's possible I've got 1 or 2 questions where you have to answer in a short paragraph. Most of them you can answer in a word or 2. Okay, but yeah, that is part of the final. You do need to take final A and final B, or just the final.
Thank you.
Okay. All right, any other questions? If not, I'll say good luck. And if I don't see you again, good luck in your education.
Thank you. Have a good night.
All right, you too.
I have a question about the unknown notes.
Uh-huh.
Do we need to wait for you to let us know if we're right about our unknown?
I'm not going to tell you until after you've submitted your unknown report what your unknown is. That reminds
Okay.
me, I do need to say one thing, and that is, people, you need to go in there and finish your unknown notes. The unknown notes are graded. It's only worth 5 points, but a lot of you have not put in the results and conclusions from your last test. So make sure you finish your unknown notes to get your full points.
In the instructions, how important is it for us to have like the date, or not the date, sorry, the time with the date for
Time is not important,
Okay, just the date.
just the date.
Okay. Whew.
Okay.
I think that's all I have.
Okay, great.
Thank
So
you.
for the unknown notes, even do you want us to submit it even if we think we figured out what the bacteria is?
You mean, do you want to state in your unknown notes what you think the bacteria is?
Well, no, I mean, like you said, go ahead and submit. A lot of you guys did not submit your conclusions.
No, I'm saying a lot of people have not put in the last results and their last conclusions for the last test they asked. That needs to be in there for full credit. You do not need to state what you think the microorganism is. It won't hurt you. You won't get anything marked down, but you do not need to do that.
Okay,
Okay.
so just basically the result of the last test
The
that we
last test
submitted.
that needs to be put in there, yes.
Oh, okay, thank you.
And the conclusions to that test, meaning
Yes, yes,
the
yes.
test tube was yellow, that's the result. And then the conclusion, the test was positive, something like that.
Okay, thank you.
Okay, all right. All right, anything else? All right, I'll say goodbye.